Hi, I'm Sean Griffin from NOAA's Restoration Center over in Puerto Rico. During the first Reef Futures meeting in 2018, back before COVID, when we had meetings in person, I presented about our plans to try and scale up some of our restoration operations by using cuttings taken from previous outplants to increase the size of the restoration footprint and to increase the number of corals at each site. Today, I'm going to present on the results of some of this work that was conducted in 2019 and 2021. My co-authors include myself, Michael Nemeth, and Tom Moore from NOAA's Restoration Center, Pedro Rodriguez, Emmanuel Irizarry, and Katie Flynn from Sea Ventures, and Hector Ruiz from HDR Reefscape. First, I want to give you some background on the work conducted at these sites prior to implementing any of the cuttings work. In 2016, Hurricane Matthew severely impacted Acropora palmata thickets in Guanica on the south side of Puerto Rico. There are about 30 acres of reef with significant damage and over 10,000 fragments at risk. There were still a lot of healthy colonies at Gilligan's, so we decided to try and transplant as many at-risk fragments as we could to other sites that no longer have healthy populations of palmata. During this work, we transplanted approximately 8,500 fragments to 10 different HMO sites, Hurricane Matthew outplant, and some of the corals were reattached in situ on Gilligan's Reef. We know the term transplant is more accurate, but we could not resist the acronym. The map here shows some of the reefs off Guanica in Puerto Rico. Gilligan's Reef is on the right, marked in red, and is about 1.2 kilometers long and has a pretty robust population of palmata. Cayo Corral is on the left and does not have as much palmata. Red circles represent some of the transplant sites, and yellow circles represent where corals were brought into nursery. Some corals were taken and transplanted to La Parguera, which is about 15 kilometers to the west of Guanica. In horticulture, a plant cutting refers to a piece of plant that is used for vegetative propagation. In other words, taking a small piece from a plant and planting it elsewhere. Here we are referring to the same thing, collecting pieces of a coral from a parent colony for asexual propagation. In the image on the left, you can see the growth from 2016 to 2019. In 2019 and 2021, we collected a small portion of the branches, less than half, from the healthy transplants. The branches were then cut into smaller pieces, about 5 to 10 centimeter cuttings, and reattached using cement. Cuttings can be used to expand the restoration footprint, fill in areas to increase coral densities within a restoration site, or transplanted to other sites to see new areas. Field teams are divided into multiple groups during the cuttings work. A team of scientists collect 20 to 30 centimeter branches from the previous transplants and load them into crates. We only harvest from healthy colonies and collect less than half of the branches from a donor colony. The branches then are cut into smaller pieces, about 10 centimeters, and placed into baskets to then be used for reattachment. Another group of divers then reattach the cuttings into designated areas. We typically aim to plant about 1 coral per square meter, depending on variations in the reef. To maximize efficiency, it's good to have 2 teams of divers working on this step. One team focuses on cement deployment, making small puddles within the designated areas, and the other dive team focuses on planting the cuttings. They go in after the first team and securely place the corals into cement puddles. Cuttings were transplanted in 2019 and 2021. We kept track of how much we expanded the different restoration sites and how many additional corals were created during this work. We also individually tagged 20 donor colonies and transplants at 4 of the sites to monitor recovery and survival. We also set aside some areas where cuttings were not to be collected to compare between areas that were harvested from it and areas that were not harvested from. We collected photomosaic imagery to look at changes in coral cover, survival, growth, and sizes at those sites. We also collected tissue samples from the tagged donor colonies for genetic analysis to make sure we have genetic diversity at the sites and that we're not creating monoclonal sites. On this slide, we can see the expansion work that was conducted in 2019 on Cayo Corral. The area outlined in purple on the forereef is the Acropora palmata zone that is less than 5 meters deep. The red polygons represent the original HMO transplant sites that were created in 2016. The yellow polygons are the new areas where cuttings were used to expand the original restoration footprint in 2019. In some of the restoration plots where mortality was higher, cuttings were used to increase the numbers of corals within those plots before expansion. The original footprint was about 3,500 square meters, and work in 2019 added an additional 2,200 square meters. Additional expansion work was conducted in 2021. These are shown in the green polygons. Some of the expansion work during 2021 involved bringing additional at-risk storm frags from Gilligan's Reef. Work in 2021 added another 3,400 square meters to the restoration footprint, bringing the total to approximately 9,200 square meters. In this table, we can see how the number of corals and the size of the restoration footprint on Cayo Coral increased from 2016 to 2021. In 2016, we transplanted approximately 3,200 palmata to Cayo Coral. In 2019, we harvested about 3,500 cuttings from the original transplants to increase the number of corals to 6,600. In 2021, we added an additional 4,600 cuttings and storm frags to increase the number of corals to just over 11,000. The restoration footprint expanded from 3,500 square meters to almost 6,000 square meters in 2019 to just over 9,000 square meters in 2021. In order to compare between the donor and control sites, we set up two permanent 10 by 10 plots within the site marked with rebar. One of the plots was designated a no-take zone, meaning that no cuttings were to be collected for areas. Photomosaic images were collected in 2016, 2019, and 2021. And we used Argisoft Metashape to create the orthomosaics. We used the images to monitor changes over time in percent coral cover, survival, growth, and size of corals using Adobe Photoshop and Tag Lab. These two graphs show how the percent cover of Acropora palmata and the average size of each colony changed over time in the control and donor areas in one site, HMO1. There are no error bars because we have not collected the last set of images from the other sites to include that data. The graph on the left shows the change in percent cover. Both sites started in 2016 with similar cover around 2%. In 2019, the donor area had a higher percent cover compared to the control area, which is interesting since harvesting had not begun yet, since it did not begin until 2019. After harvesting began, the change in percent cover from 2019 to 2021 was the same for both sites, around 5%. Coral sizes measured in maximum diameter in the graph on the right were similar between the two plots in 2016, 19, and 21. Survival rates, not shown here, were also similar. We saw 85% for the control sites and 89% for the donor in 2019 and 2021. And then overall survival from 2016 to 2021 was 73% for the control plot and 79% for the donor plot. To increase the likelihood of having good genetic diversity, at-risk fragments were collected from the entire 1.2 kilometer four reef at Gilligan's and not just one area. Initially, we sent tissue samples to Ileana Baum's lab at Penn State. In the last couple of years, we have been sending them to Eurofins for genetic analysis. The table over on the right provides a summary for the samples collected at 4 of the HMO sites. Out of 55 samples, we found 42 different genotypes, confirming that we're getting good genetic diversity at our sites. Harvesting cuttings is a viable tool for scaling up restoration efforts for fast-growing corals. For Acropora palmata, we need to wait about 3 years to be able to start collecting cuttings from your outplants. This can also be very site-dependent. As of yet, we are not seeing a significant difference between donor and control sites, but we need to collect and analyze the mosaics from the other sites. We also need to continue monitoring over a longer period, and we will continue to monitor. Some of the advantages of using photomosaics for monitoring is that we can track new fragments as they come and go into our plots. We're also able to monitor changes in percent cover and a larger number of corals. We can look at all colonies versus just a subset. Currently, the size estimates from the tagged colonies in the mosaics are similar, but we're finding differences in estimates of survival rates between tagged and mosaic data. We are looking into this some more with the next round of mosaics from the other sites. This work would not be possible without support from Sea Ventures, HDR Reefscaping, the Puerto Rico Department of Natural and Environmental Resources. Thanks to Ileana Baum's and Eurofins for the DNA analysis. We highly value and appreciate all of their support, especially Sea Ventures, who has provided most of the logistical support, not just for this project, but most of our restoration work in Puerto Rico. Funding for this work was provided by NOAA's Restoration Center and NOAA's Coral Reef Conservation Program. Thank you very much. Any questions?